All right, so I just wanted to scroll back up to the top of our DGE notebook and orient us in the workflow. So we've made our DC dataset object from the raw counts table and the sample metadata, and we've visualized the patterns within that raw counts table using PCA. So the next step is going to be to run through the 3 steps in the DESeq2 algorithm: size factor estimation, estimating gene dispersions, and performing the hypothesis test. And we'll also make new PCA plots using the normalized counts data and see how it differs from the raw counts PCA. So I am going to use the table of contents on the left-hand side here to navigate down to 2B, DESeq2 step 1, size factor estimation. Everyone please check that your notebook is still running the correct kernel, rcondenv GL4U-R, and that it says idle in the bottom left-hand side. Okay, so size factor estimation, which I just reviewed in the lecture material, is also called the median of ratios method, and it's a way to perform RNA-seq read depth normalization. So we can do this using just a single line of code. DESeq2 provides us with a function called estimate size factors, and all we have to pass to this function is the name of the DESeq dataset object where we want to normalize for read depth. We're going to run this function and assign the output to a new variable, DDS_1. So let's go ahead and run this pre-populated cell. It should run relatively quickly. And we will take a look at these normalized counts in a few steps. So that was pretty easy after going through all that math in the lecture. It really is quite straightforward to calculate the normalized matrix. Great. Next step is estimating gene dispersions. So now that our count data are normalized, we need to estimate the variance of the expression of that gene across all samples and how that deviates from the mean of that gene's expression in the data set. And we need to do this just to make sure to correct for any unexpected variability that may be due to technical artifacts. So again, DESeq2 provides us with a single function to do all of this calculation called estimate dispersions. And we're going to pass in our new DDS_1 DESeq dataset object and estimate dispersions, and then write those also right back out to the new DDS_1 object. So let's go ahead and run this cell, and we should get a little bit of pink output telling us that gene-wise dispersion estimates are being generated, the mean dispersion relationship is being calculated, and then the final dispersion estimates are calculated at the end. So to get better insight into what this step of the DESeq2 algorithm is actually doing, we can visualize the relationships between the gene dispersion or variability and the mean expression for each gene. So let's go ahead and run this function, again provided by DESeq2, called plot dispests, which simply plots the dispersion estimates. We just pass in our DDS object. So if we run this cell, we should get a plot that looks like this. If you are not seeing what I see on my screen, please let me know. But as you can see, on the x-axis are the mean of the normalized counts. On the y-axis is the dispersion. So each dot represents one gene. And you can see there's a color code here. So the gene estimates are in black. The final gene estimates are in blue and the fit line is in red. So to get a little bit more insight into our data and RNA sequencing data in general, let's use this plot to think about the following questions and answer them in Mentimeter. What is the relationship between gene dispersion and mean expression? And what might that tell you about RNA sequencing data? So I'll give you a couple of minutes to think about this one. Please go ahead and put the answer into Mentimeter when you figure it out. I am seeing some not sure in the answers, so let's go ahead and end the poll. Just give everyone a peek at the answers here. So gene disp declines as mean increases and then plateaus and increases. Not sure, not sure. Gene dispersion is how spread across the genome the genes are found, and the expression is how expressed they are. Lower expression, higher dispersion. Dispersion decreases with increasing mean expression, then reaches plateau. Not entirely sure. Yeah, so you guys are all kind of getting it at the same thing. And for those of you not sure, let's go over this together. So this is kind of, it took me a little bit to wrap my head around this. So taking a look at this plot, as dispersion, so the dispersion is highest when mean expression is lowest. So genes with mean expression tend to have really high dispersion. Remember, dispersion is variance, or how far the dispersion is, let's see, how much the variance of the expression of that gene across all samples deviates from the mean of that gene's expression in the data set. Yeah. So high dispersion for the low expression genes. Then as expression increases, the dispersion goes down until, as someone noted, that's right, it does reach a plateau. So there is a point where this relationship kind of tapers off. But especially at the lower expressed parts of this plot, we have a relationship where the lower the expression of a gene, the higher its dispersion is over the entire data set. So what does this tell you about RNA sequencing data? What I was trying to get at here is what that means is, as I mentioned before, gene expression, RNA sequencing specifically, RNA sequencing data is noisy at low expression levels. And this can be, even though a gene is biologically expressed at a low level, the chances of it then being missed in the sequencing of certain samples is higher, just because there are not that many counts of that gene. And it can get overwhelmed or swamped by other sequences in the cell. So, as if a gene has low expression, it is likely to also have pretty high variance across the data set. And so we call that noisy because that's not necessarily reliable. We have variance that's artificially inflated. All right, so then, so DSeq2 performs a fit. It kind of, remember, we talked about how DSeq2 will estimate variance or dispersion by sharing information across genes, because we know that the variance can be artificially inflated at those low expression levels. So the dispersion is estimated using maximum likelihood estimation based on other genes in the data set. So after doing that information sharing, DSeq2 performs a fit and adjusts the dispersion of all of the genes based on maximum likelihood estimation. So that's what I'm getting at in the second question. How, looking at this plot, how did the gene expression data change before, so black dots, and after blue dots, fitting on gene dispersion? So let's go ahead and answer that question in Mentimeter 2, please. Oh, and I see that when I showed the answers for the last question, it didn't refresh, so I missed I think 2 or 3 of your answers showing. So I apologize. Next time I'll make sure to refresh right before I show. So how does the gene expression data change before black dots versus after blue dots fitting based on gene dispersion? Couple more seconds. All right, let's go ahead and end voting and we can go over this together. Thank you, everyone. All right, so let's see. Before the dots are more dispersed and varied, after the clustering gets tighter, drags down the average. It fits better, less variance. The dispersions shrank. The before is spread wider, after is more tightly clustered. Gene dispersion is reduced but retains same overall shape with regard to decreasing count plateauing. Blues are obscuring blacks, correct? Yes, blues are obscuring blacks. Yep. Dispersion decreases. Okay, good. So you're all getting at about the same thing. So I like looking at this red fit line, which shows where DESeq2 is trying to fit the data to. So this fit line. So yes, the distribution tightens around the fit line. And as someone pointed out, there are a lot of black points being obscured here by blue points. So the distribution was kind of wider in terms of the black points, and it tightens up when it shifts to blue. Does everyone see that? Any questions about estimating dispersions in DESeq2? Okay, great. All right, so the final step here is to perform our hypothesis testing using the Wald test. So hypothesis testing, again, is going to tell us the probability of each gene being significantly differentially expressed in one of our groups of interest. And recall that we set up 2 contrasts. We're doing flight versus ground control. We're also doing ground control versus flight. So we're using the negative binomial model. And so the function that is provided by DESeq2 is called n-binom Wald test. And we pass in our DESeq dataset object and write out the results back to that same DESeq dataset object variable. So let's go ahead and run our Wald test cell. And it's always amazing to me that this only takes a couple of seconds, testing thousands of hypotheses at a time. But DESeq2 is very efficient. All right, so everyone's Wald tests probably should have finished by now. Let me know if it hasn't. You shouldn't get any output because we're just assigning the output to a variable. And finally, what we can do before we move on is access that normalized counts matrix from our DDS_1 variable. So remember that DESeq2 holds data in different slots. Well, one of the ways to access one of those slots is using the counts function from DESeq2. You can pass in, remember we did this before, we just passed in our DDS variable. Well, you can also pass in another argument, normalized equals true. So we're trying to access just the counts data, but just the normalized ones. There are 2 slots now. There's the raw counts data that we put in there. Now there's an additional slot, the normalized counts data from running median of ratios, size factor estimation. So we're accessing that, and then we're turning it into a data frame using the as.data.frame function, and we're assigning that to a variable norm counts. So that will become important in just a minute when we make PCA plot using these counts data. So let's go ahead and run this cell and then answer a couple of questions. So I went over a couple of hypothesis testing methods in the lecture, t-test and wald test. Here, for DSeq2, a little bit of homework looking into this vignette that tells instructions about DSeq2. Is the wald test the only type of hypothesis testing method we could have used here? So go ahead and take a look at that documentation and let me know what you think in Mentimeter. Let's go ahead and stop the voting. Thank you so much, everyone. So kind of a unanimous no. And some people noted we also could have used the likelihood ratio test, or LRT. So let's go ahead and I'm going to go to this documentation and go to the likelihood ratio test. So yep, everyone was right. DSeq2 offers two kinds of hypothesis tests, the wald test and the likelihood ratio test. Some details about what the likelihood ratio test does. And this sentence here is a hint for the next question. So I'll just kind of go over this instead of having you do the next question. The likelihood ratio test, based on its design, is useful for testing multiple terms at once. For example, testing 3 or more levels of a factor at once, or all interactions between two variables. So here, the next question was, why did we choose the wald test instead of the hypothesis, the likelihood ratio test? We only have 2 factor levels here, or 2 groups that we're comparing in either of our contrasts. One contrast is flight versus ground control, the other one is ground control versus flight. Either way, there are only 2 groups. The likelihood ratio test is a good idea to use when you have 3 or more groups, like if we had flight, ground control, and vivarium or something like that. Here we only have 2, so we can stick with the wald test. Make sure I didn't want to cover anything else. I don't think so. All right, so that's why we chose the wald test. We only had two contrasts to compare in each group. Does that make sense to everyone? In particular, the person who asked to review the advantages and disadvantages. Just want to make sure this is clear to everyone. All right. Team would have to go back. Yeah, so I have a message. If using LRT, would we have to go back and alter the way we construct the DSeq object? No, but we would have to specify that we wanted to run the likelihood ratio test when we do the hypothesis testing. So here we used this n binom wald test function. I'm sure there's one that goes like LRT or something like that.
I think maybe they were thinking about how we put like as a function of condition, or we have to put more into that part.
Yeah, I was actually just thinking about that too, Mike. Thank you. I was thinking that we would naturally be constructing our contrasts differently because there would be 3 groups that need to be compared, at least 3 groups between them. So somehow I think that the contrasts would change. But I don't think that we have to like necessarily do anything different when we create the DSeq data set. Or where did we do that? Our DSeq data set object would still just take in the counts. The sample table, I think if we were going to compare on multiple conditions, we would have to specify that here though. Yeah.
Well, this is a good example of not remembering everything. and having to dig into things when you have a specific use case.
Yeah, exactly. Yeah, exactly. Um, I don't know this because I haven't done that before, but I'm sure I could figure it out with the DC2 documentation.
So in that column, in the sample table, the condition column, we only have two unique conditions. But if you were to label samples with different conditions, so there'd be more than two unique conditions, then that would let the function know that you can do the likelihood ratio test, and it would consider all of those different groups based on the column that you designate in that DDS command.
I
Thanks,
think that
Amanda.
also gets into the difference between multiple and one factor or having multiple factors too, which is at play here, which would mean multiple columns here.
So, Amanda, not to derail, but so for GeneLab, there's always more than one factor comparison. So do you use LRT then?
That is one of the values that we generate. So you will notice we didn't do it here just for simplification. We only really have the two groups. So we do any study that we have on GeneLab, all the pairwise comparisons that we can make, we do them pairwise in both the forward and opposite directions, like we're doing flight versus ground and ground versus flight. We would do that for all groups in a pairwise manner, and that would generate the Wald statistic for that for each one of those pairwise comparisons. But then we also generate the likelihood ratio test that compares all groups as well. So you'll notice in our DGE tables, if you download them from the GeneLab repository, you'll see an LRT column, and that value would be the likelihood ratio test value that compares all the groups in the study.
Got it. Thank you. All right. Any other questions about this? All right, great. So now that we have normalized data, we've also done the dispersions and the hypothesis testing, and we'll get to the results of the Wald test in a minute. But before we get there, let's look how normalizing the data changed the global gene expression. So again, we're going to use PCA for that. We're going to use the norm counts variable that we just right here assigned, where we accessed the normalized counts from our updated data, a DC dataset object. We assigned it to this a variable called norm counts. So we're again going to take the binary log. We're going to add our little pseudo count here, and then assign that output to a new variable called exp underscore norm, and pass that into our PR comp function, which is going to calculate the PCs. So let's go ahead and run this cell, and then we can run the next cell, which again uses autoplot to create a PCA plot without sample labels, and then uses ggSave to save. All right, so taking a look at this plot, and if anyone does not see the plot the way it appears on my screen, please let me know. Taking a look at this plot and the plot that we made before with the raw normalized counts data, let's take a look at the following questions. So, is the percent of variance explained by PC2 more or less after normalization? And does this tell you anything about what was contributing to the differences in PC2? So go ahead and answer on Mentimeter, please. I'll give you a couple minutes to think about this. Look at the original PCA plot we made to look at the percent of variance explained by PC2, which is always listed here on the y-axis. So is the percent of variance explained by PC2, is it more or less here after normalization? And Based on that answer, what does that tell you about what could have been contributing to the differences in PC2? Let's go ahead and close voting. Thank you so much, everyone. So let's take a quick peek at the answers that we got. So less gene dispersion, less after normalization. Some variance was due to more sequencing depth. PC2 went down from 13.6% to 11.5%. Some of the original contribution of PC2 was an artifact that was normalized out. I feel like there's more. Less, it indicates that redepth partially explained the variance in less PCA. Percent of variance accounted for by PC2 decreases after normalization, suggesting that differences we initially observed were from differences in sequencing depth. More for flight and less for GC, not sure how to explain it. Yeah. Okay, great. So let's remember, right? So PC2 here is explaining 11.51% of variance. What about in our original PCA plot? PC2 was explaining 13.59% of the variance. So comparing these two plots, what we can see is that the percent of variance explained by PC2 has decreased after normalization. So if all we did, we normalized the data, and now PC2 has shrunk a little bit in terms of how much variance is captured by that principal component. What this tells us is that what was contributing to the differences in PC2 was something having to do with differences in sequencing read depth, or basically technical artifacts in the data. So PC2 was, our technical variation dimension. Whereas, remember, we saw before, PC1 was mainly a variance that is explained by biological differences. So PC1 is kind of our biological dimension. PC2 was our technical dimension. So it's nice to see that that it has decreased after normalization. Now, someone already kind of got at this in the answer to this question, but the second question is, look at the within group variation and compare it with the last plot. How has the within group variation and relationships changed? And why do you think that would be? I'll go ahead and put your answers on Mentimeter. Great. I'm going to go ahead and stop voting now, and we can go over this together. So, all right, I think the normalization really helped. Me too. The GC is tighter, flight is more dispersed. It's grown tighter, it's clustering as well as lower in relation to PC2. Yep, greater axes have increased. GC, all very close. Flight has more variance on PC2 and looks like in pairs. Experimental duplicates, yeah. GC is more tightly clustered. GC, yeah. So you guys are all kind of getting at the same thing, which is great. So if we go back and look at the original plot before data normalization, so we can see that the ground control samples were a little bit more dispersed. They weren't so tightly grouped as they are after normalization. And then also, as someone pointed out, the patterning of the flight samples changed. So here there are like 3 up top and then 3 coming down the side here. Then after normalization, the flight samples seem to be grouped in pairs. So what part of this question was, why do you think this is? Well, it seems like adjusting for sequencing depth helped the ground control samples cluster a little bit more tightly together. And then I don't think we're exactly sure why the flight samples are grouping like this, but yeah, there's a good hypothesis about like maybe these samples were processed in pairs or something like that. So most likely something technical rather than biological. All right, and then finally, how has the percent of variance explained by PC1 changed? And what does this mean? Great. Thank you so much, everyone. I'll give just a couple more seconds to wrap up their answers. All right, nice. 9 answers. Thank you so much, everyone. Okay, let's go ahead and stop voting now. Let's look at the answers here. So PC1 contributes to more variance, increases, yep. Further supports that differences observed in the data are due to biological factors. The percent is slightly higher. Trend stays the same. Variance PC1 increased, increased, increased. One smaller, decreased. All right, increased. Okay, so let's go over this together. So if you take a look at the percent of variance explained by PC1, that's indicated here in parentheses. So we're at 20.55 after normalization. Prior to normalization, PC1, remember, explained 18.16% of the variance. So after normalization, PC1 now explains a higher proportion of variance than before normalization. So what that means, taking a look at this plot again, is we said PC1 is kind of the biological PC. The samples are separating by condition for the most part along PC1. So the fact that PC1 has increased after normalization means that whatever we did with normalization, it served to uncloud or clarify some of the biological signal in the data. So this is a good thing. After normalization, we see PC2, technical principal component has decreased. In PC1, the biological principal component has increased. So this is a good thing. This is what we want to see after normalization because it means that we have removed technical artifacts, which has served to clarify the biological signal. So one other thing that these PCA plots are really useful for after normalization is for identifying potential outlier samples. So for example, if in this plot we noticed a sample that were way off from the other ones in its own group, you might start thinking, well, something strange might have happened with the sample. Maybe it was handled differently, maybe a mistake was made. So it's another good way to kind of get to know your data, understand what's going on, and look for potential issues. Is there any questions that you have about what we just did here? This is clear to everyone. Thumbs up. Thank you. Oh, thank you, Joe. Thank you for the thumbs up. That's great to see. All right. Okay, so in the next couple of cells, we have an opportunity for you to do a little bit of coding. So first of all, the first challenge is list the number of principal components after normalization. And in the hint, you can use the summary function we used in step 2a, but you have to find the correct variable name for your PCA. And then number 2, recreate the PCA plot, but play around with these parameters, label size, alpha. Remember, alpha has to be between 0 and 1, that means opacity. And just make a different looking plot, playing around with those terms. So you have a couple of extra code blocks here. And yeah, for number 2, another hint, first copy the auto plot command from the previous code block and change the parameters. So right here. So go ahead and play around with this code, and you can put the code that you come up with into Mentimeter once you're done. answers so far. So in the interest of time, I'm going to go ahead and work this out on my screen. And if you're still working, please have at it. But to view the number of PCs after normalization, again, we can use the summary function that we used previously, and all we have to do is pass in the variable where we calculated the PCs. So going back up here, we calculated the principal components on the normalized data and assigned the results to a variable called PCA underscore norm. You see that here? So I can just grab that variable and look at the summary. And as some of you noted, there are still 12 PCs. So I've got a bunch more answers now. Thank you so much, you guys. All right, so then we can do all kinds of cool stuff with this autoplot command. So I'm going to try some of the ones that I got in Mentimeter. All right, so let's see. Here we're running autoplot, and our original label size was 5, and there are no labels here, so that doesn't matter. But the size of points was 4, and base size was 16. So here, the size of the points is 8. Oh, and we originally set alpha to 1, so here alpha is 0.5. So let's see how this changes. Nice big plot. And I think, yeah, so either the size of the plot or the size of the font has changed. Yes, I think it's the size of the font using base size here. Nice big points using size equals 8, and they are pretty and see-through using alpha point equals 0.5. Nice. All right, what about this one here? Here's another one where size is 25 and alpha is 0.2. Oh, and they're labeled. Oh, this is so cute. I really like this. They're like bubbles with names in them. Never seen anything like that before. Very nice. All right, one more here. So smaller size and alpha of 0.5. Very nice and tidy little points that are see-through. Very nice. All right. Okay. Did anybody have an issue or an error, or have anything that they wanted to share while playing around with this code? I'm going to put the code that I just tried into the chat, so you guys can run it. I have a question privately. What is alpha doing? Alpha is changing the opacity of the points. So in our original plot, alpha was equal to 1, which means that these points were perfectly opaque. And if there are 2 points overlapping, you cannot see the one that is behind the other one. Whereas here, for example, alpha set to 0.5, that means that each point is halfway see-through. So you could see these 2 points overlapping, and you can see the one that's behind the other one. Make sense? Awesome. Any other questions or thoughts on playing around with visualizations? All right. Great. All right. So moving on, part 3 here. So we've done the DESeq2 analysis, and what that means is we ran the wald test, and I've said verbally a few times that from running DESeq2, for each gene, we're going to have a wald statistic, we're going to have a p-value, we're going to have an adjusted p-value, and we can also calculate several other metrics. So that's what we're going to do now. We're going to build out a DG output table. So this is one of the processed outputs for RNA sequencing data at GeneLab. So we're going to build out this table using the outputs from the wald test. So we have pairwise comparison columns containing flight versus ground control statistical data from the wald tests, and then we're also going to perform our own set of calculations to generate additional helpful columns containing, first of all samples. So we're going to generate a table that has our counts matrix, and then along with a bunch more metrics attached to it. So group means, standard deviations, and then at the very end, we're going to add gene annotations. All right. So the first thing we're going to do is let's go ahead and just start our table by creating a new variable called DG output table that's going to be with us for the rest of the notebook. And we're going to just start it by bringing in our normalized sample expression values. So all we're doing right here is we're taking the data from norm counts, that variable that we set up by grabbing the normalized counts from the DDS object. And we're taking all the data there and we're creating a new variable that also has all that same data. So really simple, just starting this table with the normalized counts for each sample. And let's go ahead and take a look at the first 6 rows of the normalized counts table, see what we're looking at. So the first 6 rows, note that the table currently has 12 columns, so 1 for each sample, and the rows are genes, so they're currently mouse ensemble gene IDs. So the next step is to iterate through all the wald tests to generate the pairwise comparisons of all of our groups, so the contrasts that we originally gave to DESeq2. Let's take a look at those contrasts. Again, remember we're doing flight versus ground control and ground control versus flight. So we're going to go ahead and iterate through each of these contrasts and grab the results from each contrast from the wald test results. So wald test reports log 2-fold change, wald statistic, p-value, and adjusted p-value when we get all those data. And there's some code. I'm not going to get into the details here, but just note our the function that we learned about in the R intro notebook, cbind. We're using that to take all of the information from the wald tests, which we're holding in a temporary variable called res_1. We're binding that together with our existing DG output table. So we're adding columns by using cbind. Remember, cbind adds in new columns to a data frame. So we can use cbind to update our DG output table. So we're cbinding 2 data frames together, one of which is the DG output table, and we're reassigning the output back to the DG output table, so we're updating it every time. So let's go ahead and run this cell here, iterating through the results of the wald test and grabbing the columns we're interested in and merging those back with the DG output table using cbind. So now we'll look at our DG output table in a minute, but first we can also calculate mean expression and standard deviation across all samples and add those as new columns in the DG output table too. So recall that we can add a new column to a data frame using the $ operator. So we're adding a column called all.mean and another column called all.standard deviation. And we're calculating row means, which is a function that does exactly what it sounds like, all across the norm counts matrix. So we're going back to norm counts matrix because we've just added a whole bunch of stuff to our DG output table that isn't just counts data. So let's go back to the norm counts matrix, calculate row means, and add that in as a new column. And then we're also going to calculate row standard deviations using another handy function called row SDS. Let's run that cell, runs very quickly. And go ahead and in the next code block, use the head function to take a look at the DG output table. Once you've done that, in Mentimeter, the next question is how many columns did we add to the DG output table? We started with the original 12, remember? Just the counts data for the samples. Now what have we added? How many columns have we added? Let's go ahead and stop the voting and go through this together. So I got a lot of 10s, 112 and 12. So let's take a look at this together. If I use head, remember all you have to do is pass in a variable that holds a data frame to look at the first few rows. So head DG output table. All right. So remember that we originally had 12. Thank you, Joel. I appreciate the clarification. All right. So we originally had 12 columns. And then after we look what we have here. So we have our original 12 columns, the 12 samples and their counts. Then for each gene, we've added another column here, which is the log fold change between flight and ground control for each gene. The stat column, which is the Wald statistic, p-value and adjusted p-value. So these first 4 columns are for our first contrast, right? Flight versus ground control. The next 4 columns are then for the next the other comparison, ground control versus flight. So again, log fold change, Wald statistic p-value, adjusted p-value. And then we also calculated the mean across all samples and the standard deviation across all samples for each gene. So I just want to double check that the person who said 2 columns does in fact see the 10 extra columns, because that could be an error in the code or something like that, like certain columns not being added for whatever reason. So feel free to put in the chat, or you can send me a private message if you're more comfortable. So everybody should be seeing a total of 22 columns, and it's easy to check that by going up to the top here where it says a data frame, 6 by 22. So it's 6 because the head command in R shows 6 rows, and then 22 for 22 columns. So if anyone does not see this, please let me know. Alrighty. And then finally, what is the average expression of this gene that ends in 31 across all samples? Take a look at the table and let me know in the Mentimeter. I'm going to go ahead and let's close the voting now. So mostly got the same answer, and this other answer is probably someone just looking at the wrong row. Easy to do in a table like this. So if we take, this is the 3rd row down, go across to mean across all samples, That's right here, all dot mean. And here's that other answer. That's just the standard deviation, not the mean, but it is for the right gene. So, all right, so hopefully you can see we've added a bunch of useful information to this DG table. All right, so let's go ahead and start this next big cell running, and then I'll go through it because it takes a while to run. So the next thing we're adding to our DG output table is we want to perform mean expression and standard deviation calculations for each gene across all the samples within each group, and add group means and standard deviations to the DG output table. So what we just looked at is average across all samples. Here we want to do these calculations within groups. So what this syntax is doing, there's quite a bit going on here, but first we're breaking down the, we're going again back to the norm counts. And the reason we're doing that is because we're doing these calculations for each gene across each group, flight and ground control, and we've added all this stuff to the DG output table. So we have to go back to norm counts because that just has the counts. So, sorry, I'm just helping with troubleshooting on the side here. Is everyone's code running okay? And by that I mean you should have an asterisk within some brackets here, and it's fine if your kernel says it's busy because it's just running. Okay. Thank you for the thumbs up. Yeah, so the reason that I asked you to start this code block running is because it takes the longest to run, as Amanda says. And I wanted everyone to get a head start. So mine just finished, which means that I now have a number instead of an asterisk, and my kernel says idle. So that kind of goes together. When nothing's running, it says idle. When it's running, you have an asterisk, and it says busy. All right, so I was saying we're going back to our norm counts table and breaking everything down by group, which means flight and ground control. And then we're calculating group means and group standard deviations using some R magic that I'm not going to get into right now, but I just want to point out we are adding new columns. So we're going to add new columns that say group mean and then the name of the group, and we're going to have columns that say group standard deviation and again the name of the group, so we know what we're looking at. And we held all of this new calculation in temporary data frames called group means and group stdev, and we had to use cbind again to combine those temporary data frames back in to the DG output table, and we reassign that output back to the DG output table. So rewriting that, updating that variable, adding new columns to that same table. All right, so take a look at the DG output table now. And you can use head like above and answer the following questions. So some people's kernels may be still running. Whose kernels are still running? Maybe they still say busy, and make sure if it says busy that you also have an asterisk. Okay, thank you. Anyone else? All right, so multiple people. still have their kernels running. All right. Let me know when yours finish. In the meantime, let's just do these questions together on my screen. Since I don't know how long this is actually going to take on your end. So remember, I can use head. Look at the first 6 rows of our new and improved DG output table. So if your kernel is still running, just follow along on my screen. We'll do this together. So how many columns have been added? All right. So if I scroll over here, I see the first 12, which are just the samples and their counts. Then I have the 4 statistics for flight versus ground control, the 4 statistics for ground control versus flight, and I have the all mean and the all standard deviation columns. So over here at the very end, we've added in 4 more columns. The group mean for flight, so this is the mean of each gene across only the flight samples, and similarly across only the ground control samples. And we did the same thing for the standard deviation across all the flight samples and ground control, and added those in as the last 2 columns here. So, spoiler, the answer to this question is we've added 4 columns. And then let's go back to our favorite gene here, the one that ends in all these zeros and 31, and investigate the average expression. Remember, we said the average expression of this gene in all of the samples was 94,000. So remember, this gene is 3rd from the top, right? So expression across all the samples is 94,000. So what's the average or mean expression of this gene in the flight group or the GC group? Let's see here. In the flight group, it is 76,000. In the ground control group, it is 111,000. So this gene overall has much, much higher expression in the ground control group than the flight group. Kind of interesting. Okay, so are there people still waiting for the mean expression and standard deviation code block to run? Or did it finish? Alba and Mark, did your code blocks finish? No.
Maybe I should just reset the kernel and redo things.
Does seem like it's been running for a long time. I don't know. Go ahead and restart the kernel. Yeah, I think so. Sometimes it just gets stuck. What about you, Elba? All right. So Mark, when you restart, as you know, you're going to have to run all of the cells all the way down to the 3B header, all right? Great, okay. So I'm gonna slowly move on, and if anybody else is running into issues with the kernel, let me know. Okay. All right. So if we take a look again our DGE table we've been building here. A lot of useful information. The first column, or our row names, are genes listed as ensemble mouse gene IDs. So this is great because ensemble is very comprehensive, but it's not very human readable. Doesn't mean a lot biologically speaking, unless you have a list of which gene each ensemble ID represents. So gene names are also kind of long, so it's nice to additionally have gene symbols. And then depending on which downstream analyses you want to run, there are other databases such as RefSeq, Entree, and GoSlim, and having those IDs is very useful, sometimes even required. So in this step, we are going to add a bunch more columns to our DGE table to map these ensemble gene IDs to other gene identifiers. First of all, we're going to define a new variable called organism, and we're going to set that equal to the string mouse, all in caps. And this is something that we do so that we can run this process easily multiple times using different data sets. So all we have to do is set the organism equal to whichever organism is in this data set, and then we use that to query the databases for the genes, because genes change. The gene annotations change based on the organism. So here we're setting equal to mouse, which is not necessary. This is kind of something that we do for reproducibility. So we then are going to read in using our old friend read.csv and file.path. We're reading in a file that Amanda set up for us called organisms.csv. This is a table that has been pre-populated defining the gene annotation databases needed for each organism. So you can go ahead. Oops, I also forgot to run the cell above it. So everyone, please make sure to run the cell that assigns the mouse string to the variable organism, and then run the cell defining the organism table. And we can use the code block below to view the organism table that we just imported. So go ahead and use this code block, view the organism table, and in Mentimeter, what are the first 6 organisms listed in this table? All right. All right, I'm going to go ahead and we can stop voting now. Yep, same answers overall. Human, mouse, rat, zebrafish, fly, worm. Just a note that if you spell it this way, your bioinformatics is not going to be pleased. All right, so let's take a look at organism underscore table here. So this is, as I mentioned, a table that's already been set up. As you can see, this is why we defined organism in all caps, because we're later going to query this table to get the right annotations database. So this table is set up to map the name of an organism to the official name of the species, taxon, and then an annotations database from R that has all of these gene identifiers held in that database. So we can map the name of the organism we're working with to the specific annotations database that we want to access. All right. So I kind of already gave this away. What is the annotation database used for mice and for humans? So the human annotations database name is right here, org.hs.eg.db. And then for mice, this is the one we're going to be using today, org.mm.eg.db. So these are annotations databases set up in R for gene annotations. All right. So let's go ahead and work on creating a gene annotation table. which is going to contain various gene identifiers that correspond to the respective ensemble IDs. So the first thing that we need to do is access that annotations database that I was just talking about. So as you can see, nowhere in this code block do we actually refer to this package by name, and that's because this code is set up to be organism agnostic, right? So we want to be able to, like at the very beginning, no matter the organism that we're working with, we don't have to change a lot of code. All we do is we reset this organism variable. So if we were working with rat, we would just set organism to rat, and then all of the code beneath here would still work because we have a variable named organism. We then access the organism table under the name column using the $ operator. So under the name column, we grab the row that has the name that we just set organism equal to. So we grab that row, and then within that row, we go to the annotations column, again using the $ operator, and we get that annotations database. So that's our annotations database, and we load in that annotations database using the library function. So again, just to highlight, the point of setting up this organism variable is so that we don't have to actually come down here and type in which annotations database we want to use every single time. We have a table, a lookup table. We create our variable. We look up the correct annotations database for the organism saved in that variable, and the code can continue. So the only manual thing we would have to do is set up this organism variable. So this is something that we do because this pipeline is designed by GeneLab to be able to process databases, data sets without a lot of manual changes to the code. All right, so let's go ahead and run this cell. As you can see, we get a little pink message, loading required package, org.mm.eg.db. That's what we would expect if we ran library. And then let's go ahead and run the next cell, because I think it might take a minute to run. And then, okay, it only takes a second, but I will explain it now. Okay, so we loaded the required annotations database, and now we want to use it to map the ensemble mouse gene IDs to other gene types. So we first start building out a new temporary data frame called NOT to hold all of our annotations. And I will get into the rest of this code in this code block, because I think it's going to take me more than a minute to get into this, since it is now time for a break. Thank you so much, and we'll see you back here in 20 minutes at 1:50 p.m. Pacific time.